Learn about enzymes, and before we start I would like us to look at the key words that we need to remember for enzymes. Okay, and these are the words that you should be looking to include in any enzyme exam question, depending upon what is being asked. So shape, enzymes are all about shape. The active site we're going to learn is a specific part of an enzyme. The substrate is the molecule that fits into the enzyme's active site we're going to learn. Activity is how fast that the reaction between the enzyme and the substrate is working, the rate of reaction. And we're also going to learn that it happens because of random collisions between the enzyme and the substrate. And pH and temperature, these two are two things that affect the rate of enzyme reaction. So first of all, let's look about how an enzyme works. Well, an enzyme is a biological molecule, okay, that speeds up reaction rates. So imagine if we had a molecule here that was going to react with another molecule here. We have to decide how that, or know how that would actually happen in real life. And a lot of people think, let's imagine that this is oxygen, okay, and this is glucose. Okay, sorry, rubbish. So this is oxygen and this is glucose. Now, oxygen and glucose react with each other to form carbon dioxide and water with the release of energy, and that's respiration. But do these two molecules know that they have to hit each other to react to form carbon dioxide and water? No, they don't know. Basically, what's happening is there's not just one or two of these molecules. There are millions and millions and millions of them in a cell, and they're all zooming around. They're zooming around in the cell, okay? And that's, for example, say the oxygens, and now let me do what the glucose is doing. There's millions and millions of glucose, millions and millions of oxygen, and they're zooming around. And every so often, one glucose will hit an oxygen, and they will collide. Okay, can you see here on keywords I've got collisions, and they collide, and if they collide with each other hard enough and often enough, you get a reaction. That's how all reactions happen. Okay, now the problem with biology is that we can't rely upon this oxygen and glucose hitting each other by chance alone, because it won't happen fast enough. We've got our whole body to look after. We need to produce energy which can run our bodies. So we kind of cheat. And the way we cheat is by getting enzymes which, let me just imagine if I keep the oxygen and the glucose example. Enzymes are a bit like a dating agency. They are a bit of a cheat, and they bring the oxygen and the glucose together. So if we go, so the oxygen is going to go in there, the glucose is going to go in here, and the enzyme is going to help the reaction happen by bringing them together, and we therefore get a reaction happening, and we get the products. And what the enzyme here has done is it has increased the chance of that reaction happening, so it's speeded it up. Without the enzyme, it would have just required these random collisions here, but it makes it less random by allowing the molecules that need to react to come together. Okay? Now, with that in mind, it allows life to exist in our cells at a temperature that isn't too hot, because if the temperature was too hot, our cells would die. So how does this happen? Well, we can now look at this picture. here. The enzyme is the green, and the reactant is called is the yellow. And we give these special names. The groove here is called the active site. And the yellow is called the substrate. Okay, you can see they're up in our keywords over here. Active site and substrate. And the substrate fits exactly the active site. Exactly. It's very similar to a lock and a key. And that means that the enzyme molecule is able to take the reactant and get them to react faster than they would have done if the enzyme hadn't been there. Okay? So, how does this work in an example that we've met in digestion? Well, if we look at something like amylase. Amylase is an enzyme which breaks down starch into more simple sugars, in fact more specifically maltose. Okay. So here is starch, and it's made of lots and lots of G's, which are glucose molecules bound together. And amylase here is going to take the starch and break it into smaller maltose pieces. And so it's just as we've drawn here. Here is going to be the amylase. It's going to have a very, very specific active site. For its substrate, which is going to be starch. And this starch is going to fit specifically into the amylase active site, and the amylase is going to